A very big hello to my cutest little champions. How are you all doing? I hope all my dear children are doing absolutely great, and I also hope you are kind of happy seeing the name of the chapter in the thumbnail, that is Human Health and Diseases, which is yet again a very important chapter when it comes to your PU2 biology syllabus. Now, one very important thing before I go ahead talking about anything out there, and that is, if at all you are someone who have been with us throughout the journey, my dear children, if you heard about us from somewhere else, that is your seniors, or you saw us somewhere, or you felt that we were helping at some point, I want to convey a small message out there. So the thing that I am seeing over here is that we teachers are trying our best, as in to give you the best. Okay, so if at all it's your midterm going on, we think that okay, they might not have much time, which is why we should be focusing on one shot lectures rather than part by part, because that will consume too much of your time. And also, after watching the one shot videos, you should be focusing on what you call solving enough amount of questions so that you can get these type of questions on the day of your examination as well. So that is how we have designed this whole thing. But it's actually sad when I get to see that not a lot of you are making the use of it. Okay, in terms of whenever I see people watching it or people doing the homeworks, I'm not talking just about my subject, but all the subjects out there, I feel a bit sad somewhere that I think the help that we are doing is going in vain somewhere. So please prove me wrong, okay? I know there might be so many other channels also. There might be your favorite teachers out there teaching you something. Definitely, wherever you want, you can go and definitely study as well. The only thing is that you should be studying, and that's the only focus. But if at all you feel that we are helping you in some ways, definitely share it with your friends also. Okay, that's how it goes on and goes on and on. So you have to share it with your friends, and you have to let us know in the comment section, are these sessions actually useful for you or not? So that's all. And now going ahead with today's thing, that is human health and diseases. And my dear children, over here you can see from this chapter you can get 2 MCQ questions. So in one blueprint it's 2 MCQ question. In another blueprint there is only 1 MCQ question. In another blueprint there are almost again 1 MCQ question. Now if you come about long answer, that is 5 marker question, my dear children, in one paper, that is blueprint paper, it's 1 long marker, that is 1 5 marker. Here also 1 5 marker, and here there are 2 5 markers. So you can expect either at least minimum is 1 5 marker coming from this chapter. The maximum it can go is 2. Now talking about the 3 marker questions out there. So 3 marker questions, here I can spot 1, and here also I can spot 1. Here there are no 3 markers. So again, my dear children, there are chances that 3 marker question may come, may not come. Okay, now talking about the 2 marker questions, over here in the first blueprint I don't see any 2 marker. Here I definitely see 1 2 marker coming up. So again, you can expect a 2 marker coming up or not. So what is something that is fixed from this chapter? A 5 marker question is fixed from this chapter. An MCQ question is also fixed from this chapter. So your main priority should be to focus on both of this, and you will cover up everything that comes in the middle as well. So today we are going to solve a lot of MCQs, then we will be solving a few 2 and 3 marker, and more of 5 marker questions. Are we ready? I hope you are ready. If you think we are ready, click on the like button, and my dear children, make sure you are solving questions with me and you are not just waiting for me to answer the questions. Okay, now a chemical structure is given below. Identify its plant source. What is this structure of? Any idea? Any idea? So my dear children, over here in the one shot video I taught you this thing, that is, it looks somewhat like O, and O as in which drug are we talking about? We are talking about opioids, my dear children. Okay, we are talking about opioids. And what is the opioid over here, or what is the plant source of the opioid, my dear children? It is none other than Papaver somniferum. Atropa belladonna is something else. Cannabis sativa gives us cannabinoids. Papaver somniferum gives us what? Heroin as well as morphine, which are what? Opioids. And talking about Erythroxylum coca, it is for cocaine. So this is an opioid. And what is the name of this opioid if you are specifically talking about it? It is none other than morphine, my dear children. Okay, it is morphine. It looks like O. Now, Going ahead over, so the answer is Papaver somniferum. The next question, which of the following is not a bacterial disease in humans? So that means they're just asking you which of them is not a bacterial disease. So you have pneumonia. Is pneumonia a bacterial disease according to your NCERT? Yes, it is. It is caused by Streptococcus pneumoniae or Haemophilus influenzae. Now going ahead, talking about malaria. Is malaria a bacterial disease, my dear children? No ma'am, it is caused by a protozoan. Right? Plasmodium is the name of the protozoan. So that is odd one out. Now plague is a bacterial disease and diphtheria is also a bacterial disease. So in the bacterial diseases column, no, first they spoke about typhoid, then they spoke about pneumonia, and in the pneumonia paragraph at the end they have mentioned that plague, diphtheria, everything are also bacterial diseases, and also dysentery. I'm not talking about amoebic dysentery, plain dysentery is also what? A bacterial disease. So over here you have malaria as a odd one out because it is caused by a protozoan out there. And what is the name of this protozoan? It is Plasmodium. And can you tell me, okay, chalo, first homework question out over here itself. Can you tell me which is the dangerous, variant of Plasmodium which you have studied from your NCERT, the one that causes a malignant form of malaria? Okay, you have to tell me that in the comment section. So yes, going ahead with the next question that we have. One of the following is not the causal organism for ringworm disease. Ringworm, is it a worm? No, my dear children. Ringworm disease is basically a fungal disease. Why do you call it ringworm then, ma'am? Basically, the fungus structure is like a worm. It's like round, which is why it is ring, and it looks like worm is being moved around there, which is why it is known as ringworm. Now, which one of them is not a causative organism? The one that does not cause ringworm is what? Microsporum, trichophyton, epidermophyton, macrosporum. Which one is not? The answer is macrosporum. Microsporum is, so you can remember, small pores, micropores. And then trichophyton is trichoderma, trichophyton and epidermophyton. Both these phytons are fighting with each other, you can remember like that. So trichophyton and epidermophyton, both of them are causative agents, and similarly microsporum is also a causative agent, but macrosporum is none, it's not a causative agent of ringworm disease. So your answer would be D. Okay, now going ahead with the next question out there. An intestinal parasite which causes blockage of the intestinal passage and whose eggs are excreted along with the feces of the infected person is. So there is some intestinal parasite which is there in the intestine, my dear children. So it actually causes blockage in the intestinal passage, and that's not all. The eggs of this particular parasite, my dear children, goes out to the fecus. Fecus as in 2 number. Okay, it goes out to the fecus of the infected person. So which is that particular disease? Is it Wuchereria bancrofti? Well, you know, Wuchereria bancrofti is the one that affects the lymphatic vessels and stuff like that, not your intestine. Talking about Ascaris, is that the one? Ascaris? Yes, my dear children, Ascaris causes a disease which is known as Ascariasis, and over here it is going to cause intestinal blockage, and the eggs of this parasite are going to move out through the fecus of the infected person, and then it goes to the fruits, the soil and everything, and if you do not have it, if you do not wash it properly and things like that, it can easily spread to you as well. Okay, so that is the thing. That is why we say we should be having boiled water, we should be having cooked food. Okay, so leafy vegetable is very healthy, but then eating it raw is not very considered to be very much healthy for so reasons out there. Epidermophyton and microsporum, I told you, they are a causative agent of ringworm disease, which is a fungal disorder. Fungal disease, which causes what? It causes itching and everything out there. So that is totally different. So answer is B. Cool? Chalo. Now, 5th one, elephantiasis, a chronic inflammation that results in gross deformities, is caused by elephantiasis. Why do you think a disease is named as elephantiasis? Because over here, my dear children, the people who are affected with this, their leg and all, the limb, okay, the forelimb, hind limb, right? So the hind limb, usually it is like, it's swollen up like this, okay? It looks like elephant feet, which is why the name came as elephantiasis. In actual terms, it is known as filariasis, my dear children. The name of the disease is filariasis, and it is caused by Wuchereria bancrofti. Okay, it is caused by Wuchereria bancrofti. So your answer is not Ascaris, not E. coli, not Trichophyton. Your answer is Wuchereria bancrofti. And why does it causes gross deformities? Okay, so what happens is it basically affects the lymphatic vessels out there. So the lymphatic vessels are towards this particular region. They are spread throughout the body, right? But majorly the lymphatic vessels, suppose over here, they are affected. And which is why the genital organs which are over here, which is responsible for sexual reproduction, my dear children, all of it are going to be affected, and there would be gross deformities happening over here, and also in the feet region also, because of the swelling, okay, in the lymphatic vessels and things like that. It naturally causes swelling here also in the leg region or in the hind limb region as well. Okay, so that is the thing. So your answer is what? C. Now, going ahead, which of the so which of the following is affected by the infection of Wuchereria bancrofti? Lymphatic vessels, respiratory system, nervous system, blood circulation. I already told you it's lymphatic vessels. What are lymphatic vessels? Apart from your blood vessels, there are something, for example, your blood vessels such as arteries, veins and capillaries, my dear children. In the capillaries which connects the arteries and veins, these capillaries walls are very thin, which is why some amount of plasma, certain type of cells moves out of the capillaries. Where do they go? In between the cells. In between the cells they go. Now this fluid is up there. Now there should be someone to take up this fluid and go. Otherwise what will happen? There will be swelling. Right, because there are some fluid being accumulated in between the normal cells out there, and we do not want that to happen. So who is the one that collects it? It is the lymphatic vessels which collects it. And over here, this particular Wuchereria bancrofti, my dear children, it is going to cause blockage in the lymphatic vessels out there. Now if it is damaged, do you think there will be someone to collect all this interstitial fluid or the extra fluid out there? No. There won't be anyone, which is why there is a swelling that is being caused. So that is the actual thing. Fine. So your answer would be A. Now going ahead with the next question. 7th, which of the following disease is transmitted by the bite of the female Anopheles mosquito? Sorry, female mosquito vector. I changed the question. So which of the following disease is transmitted by the bite of the female mosquito vector? So we know, ma'am, all the mosquitoes which are female, that is the only one that takes the blood. Ma'am, we know about that. Now, ma'am, in malaria also it takes like that. In dengue also it is like that only. The only thing is that the mosquitoes are different. All of them are female, all of them are mosquitoes only. So, bacha, over here if you see, do you see any option like malaria or dengue? No. You see other options like filariasis, like amoebiasis, like typhoid and pneumonia. So it's very difficult for you to understand, oh my god, malaria and dengue is not there, so what will be the answer? So if at all you have attended my one-shot lecture, you definitely know it is filariasis. Why? Because this elephantiasis disease, my dear children, or filariasis is caused by Wuchereria bancrofti, right? It causes it. But who is the one that carries this particular thing out there, my dear children? Who is the vector that carries this smaller Wuchereria bancrofti all around and spread it to us, my dear children? It is the Culex mosquito. What do you call it? It is Culex mosquito. Female Culex mosquito is the vector for filariasis. So it is also a vector-borne disease. Let me tell you, make you understand one thing. This organism does not cause the disease. It is a vector. So it is like a, a ride. Okay, person who gives the ride. So who is the person over here? It is the Wuchereria bancrofti. And who is the ride giver? It is Culex. So Culex is giving ride to Wuchereria bancrofti, and the ride is until what? Human body. It comes and affects us. Fine? So your answer is A. Going ahead. 8th question, which of the following pairs correctly matches a disease and a pathogen causing it? We have to say which of them correctly matches. Okay, so typhoid is caused by Salmonella typhi. Do you think so? Typhoid is a bacterial disease, ma'am, and it is caused by Salmonella typhi? Yes. Pneumonia is caused by Haemophilus pneumoniae? No, it is Haemophilus influenzae. So it's wrong. Pneumonia is wrong. This term is wrong. It is Streptococcus pneumoniae and Haemophilus influenzae. Now, malaria is caused by Ascaris lumbricoides? No. Ascariasis is caused by Ascaris lumbricoides. And over here, malaria is caused by what? Plasmodium. Ringworm is caused by Entamoeba histolytica? No. Entamoeba histolytica causes what? Amoebic dysentery. Ringworm is caused by Microsporum, Epidermophyton, Trichophyton. So your only answer that matches is what? A. Cool? Works? Chalo. Next. Question number 9. Appearance of dry scaly lesions with itching on various parts of the body are the symptoms of, I told you, itching. Where do you see? In the fungal disease out there. Which is the fungal disease out of them? Elephantiasis, ringworm, ascariasis, amoebiasis. Your answer is ringworm. Okay, so please remember, when I saw amoebiasis, I remember there are so many diseases which have more or less the same kind of symptoms, or which has the same kind of affected organ. For example, if you talk about ascariasis and amoebiasis, both of them are causing trouble in the intestine itself. You get it? So you should understand the small differences that are there. Amoebiasis is caused by a protozoan, whereas ascariasis is caused by what? A helminth. Okay, it is an asc helminth that causes it. So the causative organism is different, the way it acts is different. Okay, somewhat the symptoms also, there might be difference in your NCERT. So read them properly. Well, if you have attended the one-shot lecture, by now you might know all the symptoms properly. So yes, going ahead, B. Next, hemozoin is what? Hemozoin, ma'am, I have heard of it. I do not know, but what is it? Precursor of hemoglobin, toxin released from streptococcus infected cells, toxin released from plasmodium infected cells, toxin released from hemophilus infected cells. Well, the answer is toxin released from plasmodium infected cells. Why is this the answer? That is the question. Because, my dear children, plasmodium, which is causative organism of malaria, no? There are different stages of plasmodium. One such stage affects the RBC. And in the RBC, what do you have, my dear children? We have hemoglobin. And you know, when hemoglobin is broken down, you get heme protein and also you get globin protein. This heme protein which is there is kind of very toxic for this particular plasmodium, which is why it quickly converts this into something else which is known as hemozoin. Remember, all of this is happening within the RBC itself. Now, this hemozoin which is there, it is actually toxic for us if it is released. So basically, when in the RBC all this thing is happening definitely by them only, and they are doing one more thing. They are doing reproduction also over here. So basically, when the number increases way too much, this RBC is going to burst open. And what is in there in the RBC? Hemozoin is there. It is going to be released. And this hemozoin, my dear children, is acting like a toxin for us. Why? Because when it comes, my dear children, our body will be reacting in the form of giving fevers, chills and things like that. Which is why we say in malaria, we get high fever and also chills. It's when the release of hemozoin is happening. So toxin released from the plasmodium infected cell is known as hemozoin. And where do you get the hemozoin? It is actually from the hemoglobin itself. The heme part of the hemoglobin is converted to hemozoin because heme is toxic for them. So they thought, let's make it toxic for the human body, not us. So that is what they do. Okay, so here is the whole entire thing. I have explained that in the one-shot lecture, but still I am telling you, plasmodium is there. It is a protozoan and it causes malaria. How? So in the Anopheles mosquito, okay, in the salivary glands, in the saliva, you have a stage of plasmodium which is known as sporozoite stage. Now, when the mosquito comes and bites us, it does not give us the blood. We give it the blood, right? And for it to easily suck our blood, it releases certain enzymes through the saliva. Okay, so when it passes the saliva, my dear children, what happens? It also passes the sporozoites out there. Now, these enzymes are known as anti-clotting enzymes because they do not want the blood to get clotted. Our blood usually gets clotted, no? If at all there is a wound and all, that will get clotted so that the blood is not wasted. So, they give anti-clotting enzymes so that they can easily drink and go. So, while they do so, since there is a salival transfer out there, sporozoites move into the human body. And in the human body, they are the terrorists, right? The first camp is there in the liver. And the cells of the liver are known as what? Hepatocytes, not hepatocytes, it is hepatocytes. Once they get into the hepatocytes, my dear children, they will undergo asexual mode of reproduction. Okay, and then when they are in too much number, the hepatocytes will get lysed, as in they will get break open. Okay, and then, my dear children, all of whatever content was there, it goes to the blood now. In the blood, they enter the RBC, my dear children, which is the second camp, okay, which is their second camp. Again, they do the same thing, asexual reproduction. Now, the RBC gets lysed, and what will happen? As I said, hemozoin will be released, which causes what? Fever and the chills. Apart from that, while I say that asexual reproduction is happening, my dear children, a lot of different forms are occurring, okay. One such stage reaches, which is known as gametocyte stage. So, initially, it was sporozoite stage, now it is gametocyte stage. It is formed in the RBC itself. Okay, these gametocytes are the ones that can in future form the gametes. They are not going to form the gametes in human body, please remember that. Okay, gametes are not formed over here. In human body, there is no sexual reproduction of plasmodium that is happening. Okay, not even the gametes are being formed. Gametocytes are being formed, and these gametocytes, my dear children, again when the mosquito will come and bite, they are taking the blood, and the blood has what? Gametocytes. So they will take this gametocytes, and now the mosquito, in the mosquito's gut, no, these gametocytes will give rise to what? The gametes out there, and the gametes will be fusing and forming zygote in the mosquito's gut. It's none of our business. But anyway, whatever damage it had to do, it already did. So please remember, sexual reproduction of Plasmodium does not happen in our body, it happens in the mosquito's gut. Okay, and in what stage does it enter our body? Sporozoite stage. And what is the thing that causes trouble to us? Hemozoin. In what stage it is there in the RBC? Gametocyte stage. Okay, all of this thing, there are small things that you need to remember. Fine, and it is a protozoan, that's all. So your answer for this question is, it's toxin released from Plasmodium infected cells. Done? Deal? Okay, cool. Next question. Which drug is used as a medicine to help patients cope with depression and insomnia? Depression and insomnia. The insomnia means you are not able to sleep. That is insomnia, a high version of it. Not like today I am not able to sleep. It's not like that. For many days and for entire like more than 24 hours or more than that also they might not be able to. The severity depends obviously, but yes. So what is that drug which is used to treat depression as well as insomnia? Is it morphine? No. It is a painkiller. It is definitely a painkiller and a depressant, but not that. Okay, amphetamines, yes. Cannabinoids, no. Cocaine, no. Your answer is amphetamines, benzodiazepines, all of it. Okay, benzodiazepines, amphetamines, they are all the ones which are used to treat depression and insomnia, not anything else out there. Please remember, they are difficult names, but if you read the chapter properly, you will remember it. How do I remember it? By teaching it to you guys only. There is no another way to remember all these things. Okay? Now, during the life cycle of Plasmodia, gametocytes develop in. Tell me now. Gametocytes, they develop in what? Human RBC, human liver, salivary gland of mosquito, gut of the mosquito. Where do you see the gametocytes are being developed? Where are they formed? Human RBC, my dear children. And these RBCs are taken up by the mosquito which comes and wants to drink the blood. And then in their body, it is going to form the gametes. Okay, so your answer is A. So I hope now you're human liver. No. It's wrong, okay? It's RBC. Okay. Going ahead. Which of the following is a viral disease? Pneumonia, typhoid, common cold and malaria. What do you mean by viral disease? So people usually assume the viral disease sometimes is the one that you can get from one organism to another. Yes, it is, but viral disease in this sense is the ones which is caused by viruses. So which of them is a viral disease? Pneumonia, typhoid, common cold or malaria? It is what? Common cold, which is a viral disease out there. So your answer would be C. Pneumonia, as I told you, it's caused by bacteria. Typhoid, it's caused by bacteria. Malaria is caused by protozoan. Well, let me tell you, and common cold is caused by a virus, that is rhinoviruses. Pneumonia is actually also caused by viral agents as well. Okay, so it's not always by bacterial thing, but I'm generally telling you, it's like in your book, it's just bacterial, but please have a broader understanding that it's not just bacterial pneumonia, it can be viral pneumonia as well. I have got it, which is why I'm telling you. Okay, so yes. So your answer is common cold. Now going ahead, the human immunodeficiency virus HIV primarily attacks and destroys what? Red blood cells, Th helper lymphocytes, platelets, T cytotoxic lymphocytes. It is the one that helps, is the one that gets destroyed first over here in this particular story. So HIV viruses, they are going to first affect the T helper cells, which you also call as Th lymphocytes. Why? Because they are going to help in telling the other cells that hey, something has happened, you have to do something about it. So they are like the messengers out there. In some way, they are the ones which are being primarily attacked. Okay? So always remember, it is T helper cells out there. Next question. Which of the following is an autoimmune disease? Autoimmune disease means our body is not able to, our immune system is not able to identify which cells are our cells and which cells are not. So basically it ends up killing both of them. You get it? So it's known as autoimmune disease or disorder. So over here, rheumatoid arthritis, tuberculosis, malaria, AIDS, all of it is given, my dear children. Which one of them is an autoimmune disease? It is rheumatoid arthritis. Okay, it's none of the other ones out there. It is just rheumatoid arthritis, which is an autoimmune disorder which majorly what do you call, affects the synovial fluid which is there in between the joints, okay, in between the joints out there. So yes, so your answer would be A. Now going ahead, the substance produced by a cell in viral infection that can protect other cells from further infection is. So basically, suppose this cell is affected by a virus. So this cell will be screaming to the other cell, which is pretty happy, my dear children, to save yourself, to save yourself. But what does this release to say that? It releases something which is known as interferon. So it is intervening between them, it is interfering, you can remember like that. So it is interferon, but it is interfering what? For a good reason as well. So your answer would be C. Not serotonin, not colostrum, not histamine. Histamine is released during allergies usually. Colostrum is the first milk which is being released, okay, by the mother while breastfeeding. Serotonin again, it would be helping in all the allergic reaction and in general reactions of swelling and things like that, okay. So going ahead, your answer is C. And now 17th question. Antibodies present in colostrum which protects the newborn from certain diseases is. So what is there in the first milk of the mother which protects the newborn from certain diseases? So always remember this thing, gamed. These are the 5 antibodies that we have. This is the first antibody that passes through the placenta from the mother to the baby. That is immunoglobulin G, IgG. Okay, antibody G. Now the second antibody which passes through the mother's milk is antibody A, and I'll give you a good way to remember it. Most of us calls our mother Amma or Ai, or you might have different names. It starts from A, right? So you can remember A. It's immunoglobulin A. M and D, the functions of it are not being put in your NCERT, so as of now you do not need to worry much about it. E, please remember, it is for allergy. E, allergy, you can remember like that. So E is being released when someone is having allergy, or allergy is being caused because in your body there is too much amount of IgE being released, either way. So your answer for this question would be B, IgA. Next question. Which of the following is not a lymphoid tissue? Not a lymphoid tissue, as in a lymphoid tissue is where my lymphatic cells are being produced, or if at all not produced, they are going to show their effect there. Two things can happen. Either they are produced there, or probably they are going to do their fighting scenes, or they are going to show their effect there in the lymphoid tissue. That is the meaning. So spleen is a lymphoid tissue, tonsil is a secondary lymphoid, both of them are secondary lymphoid tissue. Thymus, which is there, it is a primary lymphoid tissue. Why do I call it primary? Because this is where the lymphoid cells are being formed, lymphocytes are being formed. Spleen and tonsils are where they go and show their fighting scenes and everything. Pancreas is the odd one out. It is not a lymphoid tissue. So your answer for this question would be C. Going ahead, the site where lymphocytes interact with antigens and proliferate to become effector cells. So I told you there are certain regions, primary lymphoid organs, where they are just formed and they become sensitive to antigen. They become sensitive to antigen, as in if at all there is antigen, in future they might show the effect. That is what you mean by being antigen sensitive. They are not showing the effect now, but they go to some other places and they act like effector cells. What do you mean by that? Now they are totally mature enough to what you call do the fighting scenes and everything. So they are the secondary lymphoid organs. So that is being asked over here. The site where lymphocytes interact with what? Antigens, that is their dushman, their enemy, and proliferate to become what? Effector cells, as in the ones that can fight off everything out there. So what is this? The secondary lymphoid organ. So over here thymus is given and lymph node is given. Lymph node is a secondary lymphoid organ, but thymus is a primary lymphoid organ. Okay, bone marrow again is a primary lymphoid organ. Thymus is also a primary lymphoid organ. Out. Peyer's patch and tonsils, both of them are secondary lymphoid organs. Bone marrow, primary lymphoid organ. Spleen, secondary lymphoid organ. So which is the one that has both of them to be the secondary lymphoid organs? It is just the C option, which is Peyer's patch as well as tonsils. Always remember, being antigen sensitive happens in the primary lymphoid organ, that is bone marrow and thymus. Being effector cells happens in the secondary lymphoid organs, that is all of them that I discussed: Peyer's patch, tonsils, spleen, everything. Okay. Now, antivenom against snake poison contains what? When I say that, I have been, oh my god, my hair scared you. Okay, antivenom is being given. Okay, so a person got a snake bite and quickly they are taken to a doctor. Now doctor gives them antivenom. What does this particular thing contain? Antibodies. It contains antibodies. So my dear children, the venom has the antigens out there. There should be someone to destroy these antigens are there, they are the antibodies, and they are the preformed antibodies they already have, okay, that I already have with me. I'm not waiting for my body to produce antibodies. In the antivenom, there are already the antibodies been up there, so that the body immediately gets it. Okay, it's a ready-made thing that is there. So your answer is C, not A, not B, not D. Now going ahead with the next question, 21st. Vaccines work by what? Stimulating the production of antibodies, killing the pathogens directly, reducing inflammation, blocking the entry of pathogens into the body. How do you think vaccines work? So we were all given COVID vaccine, the people who got it, the disease, who didn't get the disease, still all of us were given vaccines. Why? Because vaccines, my dear children, they are, either they can be weakened pathogens or some part of the pathogen which is not going to cause any trouble to us, but just ignite our immune system. It just says to our immune system that something like this can come in future, and you would have to release the antibodies that you have released now in more amount. So whenever our body sees something for the first time, it will obviously try to save us by producing antibodies, but the primary response of our body is very less. It may not be able to overcome the disease out there. But the second time the same guy tries to come in, my dear children, our body will produce more amount of antibodies out there. It will give you a good secondary response. So we want our body to, we cannot wait that, okay, let that person be infected with that disease, he'll get the primary response, and the second time every second response. We cannot wait for that, which is why we take vaccines. Okay, so it's not just COVID vaccines. Obviously, COVID vaccines had its, own consequences. A lot of people have a lot of things to say about it. But in general also, from our childhood, we are taking polio vaccine, this vaccine, that vaccine, right? Which actually helps us not developing a disease out there. So basically, it's like giving our body a reminder that you have to produce antibodies that you have produced now in future as well. So it's just that thing, that's all. So vaccines work by what? Stimulating the production of antibodies. Okay, we're just giving the weakened pathogens or things like that so that our body can produce antibodies. And when antibodies are produced, my dear children, there are memory cells, okay, which are also being produced, and this have the memory that something like this has happened. Now the next time when it happens, I have to produce more antibodies. So this is your answer. Okay. Next, 22nd, which form of pathogen is used in vaccination? Which type of pathogen? Do I put a live pathogen inside the body of someone who wants to take the vaccine? No. Activated and strong pathogenic antigens? No, ma. Preformed antigens? No. Inactivated and weakened pathogen? Yes, such thing that they cannot get activated inside our body. Hyperactive and strong pathogen? No. So in coronavirus, you remember that, my dear children, everything was like this. Okay, so there was some proteins which were at the top of the body, my dear children, of the the viral body, which were known as spike proteins. So the elements of spike proteins were being put down in the human body such that they could react to it and they could give a response. So that is the exact thing that is being done everywhere. Some part of it, or probably inactivated and weakened pathogens, are being used out there. Next question. Heroin formed by acetylation of morphine is commonly called as what? Coke, crack, smack, charas. Okay, so heroin, which is there, opioid, it is formed by acetylation of another opioid, that is morphine, my dear children. And what do you usually call this? Coke or crack or smack or charas? It is smack. It is what? Smack. And heroin is a depressant, my dear children. Okay, it's a depressant. Fine, coke and all, you call cocaine. So yeah, hepatitis B vaccine is produced from. So in the vaccination session, at the end of that page, my dear children, a small line is given, and usually children don't see that line. So there's a question is, hepatitis B vaccine is produced from. So you know, in biotechnology, you will study in biotechnology principle and process and all the things, my dear children. You take the help of certain small organisms, and since they can divide really faster, so what do? You do certain changes in them. So, and you put something else in their body, and you make them produce that, you make them replicate that, because we want that. Okay, because we want that. So you are asked that, so over here it is hepatitis B vaccine. Okay, so that means hepatitis B, some part of it, some part of it we want it. Not hepatitis B, some part of it, basically the organism that causes it, it's some part of it we wanted to form the vaccine, which is why we made it amplify using an organism, a small organism out there, which is none other than the yeast, my dear children. It is the yeast. So inactivated viruses, no. Haemophilus influenzae, no. Salmonella typhimurium, no. Your answer is just the yeast. So hepatitis B vaccine is produced by yeast. Okay. Next, 25th. Which of the following cells actively participate during allergy? During allergy. So there is a cell which is known as mast cells. You can call them mast cells, either way. So mast cells which are there, my dear children, what happens is that they will have areas where they are bonded to the antibody, that is E, IgE, immunoglobulin E. Now immunoglobulin E would be having the antigens that are causing the problem, that is for example a dust or something like that. Now when they are attached to the mast cells are going to release histamines, okay, serotonin and all this thing, which is going to cause what? Which is going to cause inflammation, vasodilation, all of this thing. Why vasodilation? So basically what happens, our blood vessels which are like this narrow, my dear children, you want lot of other cells, immune cells to come over here, to fit in over here, to give rise to the huge response out there, which is why the blood vessels are going to get dilated when these guys are released, such that all the cells can come over here and do their action. So that is known as vasodilation, and this vasodilation, my dear children, causes what? Inflammation. This in turn causes what? Inflammation out there, which is why some people, when they are allergic reaction, they see a lot of swelling and this and that happening. I hope you got it. So it is mast cells which is causing this. Okay? Fine. Now the next question. 26. Charas and ganja are the drugs which affect what? Charas and ganja, they are cannabinoids. We get them from cannabis sativa. So cannabis sativa is leaf, stem, every single part can be used as a drug. So charas and ganja are got from cannabis sativa, and what do? All of them, they have a similar function, that is they affect the cardiovascular system, not the respiratory system, not the digestive system, not the nervous system, that is CNS, nothing like that. It is the cardiovascular system which is affected with the help of charas and ganja. So your answer for this question is what? B. Okay. Now the thing, there's one more last question, and if at all you can, please answer this in the comment section. This is your homework number 2. Which drug is being excessively taken by some sports person nowadays? Opioids, barbiturates, cannabinoids and LSD. So I'll tell you where is it. It's in your NCERT. I've given you a clue. It's your job to open your NCERT, read through the line. So where atropa, belladonna and everything is given, that is just after cocaine. Okay, just after cocaine paragraph, or actually within that paragraph itself, the cocaine paragraph itself, you may be able to find it, but I want you to read it and you to tell me that. So which drug is something that has been excessively taken by some sport person today? And I don't think I have put the answers. If at all I have put, I'll just change. Yes, and with that we end all the MCQ questions, my dear children, and now we are going to go ahead with the 2-marker question, and then we will go ahead with the 3-marker questions out there. The first question that we have in this category, and which is being repeated most of the times, is this: draw the structure of antibody molecule, which was given in the model question paper as well. So how do you draw the structure of antibody molecule? You should know what is it made up of. So this big line that you see over here, this is the heavy chain. What do you call this as? Heavy chain, my dear children. You're not able to see, I'll write it and I'll tell you. If you're not able to see, I'll write. It is the heavy chain. And now the small chain that you see over here, it is the light chain. Now we are going to get inside the heavy chain and light chain. So over here you see the color is different at the tops, but the bottom the color is same. Similarly here also it's different. Here and here the red is same, but the top it is different. So what do you call these regions in the lower regions? The lower region of the heavy chain is known as constant region. What do you mean by that? It's constant in all the antibodies, IgG, IgA and things like that. Okay, and the top region that you see over here, they are known as variable region. Ma'am, it's just there in the heavy chain? No, it's there in the light chain as well. This is also what? A variable region that you have. So it is going to be different in all the antibodies out there. And in between these, The heavy chains, you see sulfide bridges out there. So these are sulfide bridges. There are more sulfide bridges out there. But if you talk about the heavy chain and the light chain, it will be less. There will be only one. But over here, you can find multiple of them. Okay, and here also between the light chain, between the constant region of the light chain, you can find it. Here also you can find it. Here also, in between the constant region of the heavy chain also you will find it. In the variable region also you will find it. The sulfur bridges or the sulfur bonds are there over here in the antibody molecule. Now, at the top region where both the regions are variable, no? Variable region of heavy chain, variable region of light chain. This area is known as antigen binding site. This is where the antigen will come and bind. Okay. Similarly, on the other side also it is there. So this is the basic structure of an antibody molecule. It can be different in different types of antibodies. Okay, so there can be something that can have 5 of them together being pumped up. You'll study about that in later if at all you choose biology. And my dear children, one more thing. So over here, I hope you know that immunoglobulin is a protein. Okay, that is antibodies that you see over here, they are proteins, and proteins are made up of what? Amino acid. Amino acids would be having, each amino acid which be there, it has an amino terminal, that is an N terminal, nitrogen, okay, and a carboxyl terminal, that is C terminal out there. So all of them, they come together like this. So at one end you have C terminal, and now it's joining. At another end you will be having N terminal. So this is your N terminal. You can remember antigen binding site, N is there. So antigen binding site has the N terminal, and over here on the other side you have the C terminal. So this is the structure of the antibody molecule that you have to make, and it is a question that is mostly being repeated as well. Okay. Next question, list any 2 methods of HIV transmission. How do you think HIV is being transmitted? What is HIV? Human immunodeficiency virus. What does that cause? It causes AIDS, okay, acquired immunodeficiency syndrome. So what are the 2 methods in which someone can get HIV? If I give you a handshake, suppose, God forbid, if I have HIV, okay, now if I give you a handshake, will you get it? No. It does not happen like that. So which is why I'm telling you so openly, okay. You should not be, ignoring someone who has the disease. You should not be stigmatizing that because it's a, a normal disease. You just have to be precautious. That person also has to be precautious about certain things. That's all. So what are the different ways in which someone can get HIV? Sexual contact with the infected person for sure, because it is going to pass to the body fluids out there. Second, transfusion of contaminated blood and blood products. By body fluids, I don't mean any body fluids like such as saliva and things like that, okay. Okay, please understand that. So genital fluids and things like that, blood and all, through which the sexual contact, in fact, someone can get it. And now transfusion of contaminated blood and blood products, okay. Sharing infected needles as intravenous drug users. So for example, a drug user is there, they usually have injections. So I had an injection, I gave the same injection to that guy, but I have a disease and that person will also get the disease. You get it? So sharing infected needles from infected mother to the fetus through the placenta. It's kind of rare. Now we have so many medicines being popping up there, but still, if at all there is some blood transfusion happening between the child and the mother, there are chances that yes, from the infected mother it can go to the fetus as well through the placenta. Now, usually the blood is not something that is being transferred. It is all the important nutrients which it's not like direct contact of blood is there. I hope you get it. But at rare chances during delivery and doing things like that, if at all something fails, my dear children, there can be a chance that it can happen. Now, organ transplantation from an infected donor to the recipient. Again, there is a high chance. Okay, so these are the methods by which HIV transmission can take place. Please make sure you are knowing all of it, because any type of questions can come again. Now comes the 3-marker question. So the 2-marker question is only that, those 2 questions, and now we have the 3-marker question, and over here you have to be attentive because this is about what? This is about the drugs out there. So name of the drug, its source and the nature is given in the table below. Find A, B and C. So this is the name of the drug, this is the source plant, and this is the nature. So over here, what is the name of the drug which you get from Papaver somniferum? And that is a depressant. I'm not telling it's a painkiller, it's a depressant. So from Papaver somniferum you get 2 drugs, ma'am. It is either morphine or heroin. So morphine is a painkiller, okay, it's a painkiller, but if you just talk about depressant, so it has to be heroin over here. It has to be heroin. So yes, so answer would be heroin. Now cannabinoids. So cannabinoids is the name of the group of drugs. The source plant of cannabinoids is what? Cannabis sativa, and its effects is on the cardiovascular system out there. Now over here, cocaine. You get cocaine from erythroxylum coca, and what does it do? What is the nature of this drug? What does it do? It affects the central nervous system, isn't it? It affects the central nervous system. So your answers would be this. So the first, okay, the first answer is what? Heroin. From papaver somniferum, you get heroin. You get heroin. That is a depressant. Cannabinoid source plant is what? Cannabis sativa. And cocaine nature is what? What does it do? It affects the central nervous system, especially where it affects the dopamine secretion and stuff like that. So usually the dopamine, it stops after some time. The release of it stops after some time. But over here it interferes in that, which is why there is a continuous release of dopamine. And when there is a continuous release of dopamine, you feel a sense of euphoria. Okay, you feel a sense of euphoria as in too much happy, or it's uncontrollable. You might start seeing things also which is never there. So actually it might sound fun, but it is not exactly as fun as it sounds. Always remember that. So yes, it affects the central nervous system, and as I told you, it majorly affects the dopamine secretion and things like that. Okay. Now describe any 3 properties of cancerous cells. What are the 3 properties of cancerous cells, my dear children? First one, you know that a normal cell has a controlled growth. Obviously has what? A controlled growth. Cell divides into twice, and after a span, that's all. It cannot divide anymore. But over here, in the case of cancer cells, they would be having uncontrolled growth. Now not just that, ma'am. The other feature that I can relate here itself is that basically here, if one cell divides, another cell comes up, they show something which is known as contact inhibition. What do you mean by that? Basically, if one cell is coming in contact with another one, they'll stop the division. This is known as contact inhibition. When they're coming in contact, they're getting inhibited. Now over here, in this particular case, they show loss of contact inhibition. Loss of contact inhibition, as in, if at all the cells are coming closer, my dear children, even after dividing, they will keep on dividing. This is known as loss of contact inhibition. So this is another property that you need to remember about cancerous cells. Apart from that, normal cells, they stay at one place. Wherever they are needed, they just stay there. But in the case of cancerous cells, they show a unique property which is known as metastasis. What do you mean by that? They move around throughout our body. They go to different places. They relocate to different places, and again causes tumor there out there. Okay, so this property is known as metastasis. So first is uncontrolled growth. They divide rapidly without responding to the normal regulatory signals. So we all have normal regulatory signals in the normal cells, but here in this case, no. Loss of contact inhibition, they grow even when in contact with other cells, forming what? Tumors. Metastasis, they can spread to distant body parts via blood or lymph. So these are the 3 properties of cancerous cells which you need to remember. Now, name the pathogen, vector and symptom of malaria. Very easy question. What is the name of the pathogen, my dear children? Name of the pathogen is Plasmodium. What is the vector? Ma'am, vector is mosquito. If you write that, will you get marks? No. You have to write female Anopheles mosquito. And now, symptom of the malaria. Symptom of malaria is what? High fever and chills. Something that I can easily remember. High fever and chills. These are few things I can remember. Now, over here when I name the pathogen, should you just put Plasmodium? You should put the names Falciparum, P. falciparum, P. vivax, P. malariae, all of it, and tell that P. falciparum is what? The one that causes the malignant form of malaria. So pathogen is Plasmodium, P. vivax, P. malariae and P. falciparum. Always remember the second word. The species name always starts with a small letter. Okay, and tell that it is P. falciparum that causes the malignant form of malaria. Now vector, by biting the infected female, by biting of, not biting the, is biting of the infected. Don't go and bite any mosquitoes. Biting of infected female Anopheles mosquito. Symptom, high fever and chills occur on every alternate day. Vomiting and everything is also there. Okay, fine. Next question. Differentiate between active immunity and passive immunity. What do you mean that? Active immunity. Active immunity means basically making your body very active enough to react to certain things. Your body is doing everything alone. But basically, what you're doing exactly over here is that in vaccination, what do you see? Sorry, not to make it too much, confusing for you guys. I'll come back to the topic again. What do you exactly mean by active immunity? Over here, you want your body to actively participate and make the foreign particles go away. So how do you do that? Over here, natural infection. When a natural infection happens, suppose I have got rhinovirus in my body. My natural reaction to it will be about producing of antibodies, production of white blood cells, and a lot of other things are there that causes the reaction like cough and cold and a lot of things. Okay, so over here, natural reaction is making my body to actively participate in the immunological response out there. The second way I can do this is vaccination. In vaccination, I am going to give you what weakened form of pathogens inside my body, and now again my body will react to it. Anyways, it's my body which is actively participating in the immunological process. This is known as active immunity. Now when I talk about passive immunity, it will be all the more clear for you. Passive is where you're not making your body react to anything. You know, oh my god, your body is so weak, I cannot do anything with it. So what you're doing, you're giving ready-made antibodies. Okay, ready-made antibodies to the body so that it can save these ready-made antibodies can save our body in some ways. So in a baby, okay, when a baby is being born, I told you from the mother, the first milk contains what? Immunoglobulin A. Why do you think mothers give the the antibodies to the babies through the milk? Because the baby itself does not have the capacity to produce any antibody as of now. It's in a growing stage. So what do? You're giving antibodies. So this type of immunity, okay, which you're getting from somewhere, this is known as passive immunity. Now you tell me, okay, active immunity, you're making your body participate in that and it is producing antibodies. So do you think it is going to be long living, this immunity that you're getting? Do you think it will be there somehow next time also it will protect us? Yes, it does. But in the case of passive immunity, once these antibodies are being utilized, it's been done, my dear children. So this is a temporary immunity that we are giving. Okay, so passive immunity is for a temporary period. So there are small things which are there, and I'll show you now in the form of definitions as well. So immunity developed due to injection or of exposure to okay, the immunity developed due to what? Any injections, okay, that is in the form of vaccines or things like that, or what? Exposure to some infection. Now, passive immunity, the immunity developed due to ready-made antibodies. Done. Now over here, it is slow and takes time to give its full effective response. That is there, it is slow, but it is what? It is giving you lifelong immunity as well. Over there, you're not waiting for your body to react. You're immediately getting it. It's ready made. So there's an immediate response over here. It provides what? Temporary immunity. That is a problem. Okay. Now, memory cells are produced for a long-term immunity. I told you in our body, whenever lymphocytes are being produced, the B cells, B lymphocytes when are being produced, they produce memory B cells also, memory cells also. Okay, or even when T lymphocytes are being produced, they also produce memory T cells. So anyways, the memory of something has taken place is there. Memory cells are there. They can help in giving us immunity later on. Okay, memory cells are not being produced over here because there is no active participation of the immunological system out there. Now, I'll tell you a question and you give me the answer. If at all someone comes with a snake bite, okay, I told you, you give antivenom. What type of immunity is this? Active immunization or passive immunization? Do you wait for that person to produce antibodies by themselves in their body? No. You are going to give ready-made antibodies. So that is a form of passive immunization. Okay, so anti-venom, tetanus injection, all of it is what? Passive immunity. You are giving ready-made antibodies at that particular point of time. I hope you got it. Antitoxins. Okay, antitoxins, everything comes under passive immunity. Fine. Now, one more thing is there. Define allergy. What is allergy? Allergy is what? Overacting. That's what I do most of the times. Yes, there might be so many people agreeing to that, but that's my way. But yeah, so allergy is what? The exaggerated response of the immune system to certain antigens. it's a not normal way. It's an exaggerated, which is why we have swelling, we have this, we have that. All of us, probably you, me and someone else, might be in a room full of dust. I may not react it the way just as you do, or probably I may react it in a very bad way rather than you. I'm not talking about mental reaction, I'm talking about in a way my body shows up. My body might have certain bumps being formed. I might just start coughing, I have cold and this and that. So the exaggerated response, response of what? Our immune system to certain antigens or allergens, my dear children, in the environment is known as allergy. Which is the antibody responsible for it? Antibody E, allergy. Which are the cells responsible for it? Mast cells. Fine. Now, mention the type of carcinogens with the example for each. What do you mean by carcinogens? Carcinogens means cancer causing things are known as carcinogens. What are the different type of carcinogens that you can recall, my dear children? Race, okay, biological carcinogen, physical carcinogen, chemical carcinogen. For example, someone is a smoker. Now a smoker, when he smokes, he has nicotine. In the tobacco, it releases nicotine, and nicotine can cause, my dear children, what can cause? It can cause cancer. Okay, or the smoke of it can cause what? Lung cancer. So this is what? A chemical carcinogen. Fine, it would be chemical carcinogen. Now physical carcinogen, for example, I am being told that you should not be keeping mobile at the near your body when you are sleeping, Jasmine. So I'll say, why can't I use mobile phone at the night? I want to chill. So the answer comes like, you can get cancer, but we do not know how do we get it. So basically the rays which are being released over here, my dear children, by your mobile phones, all the electronics out there, even the sun, the UV rays which is there in extra quantity, my dear children, all of it can be troublesome. It can cause cancer in our body. One cell, okay, one small mutation in a cell can cause what? Cancer. And I'll tell you, in our body, when I am talking, when you are listening, a lot of mutations are being happening. But there is a DNA repair mechanism also in our body which repairs all of it. You get it? Sometimes it goes out of sight, which is why our body would not be able to sense it, and that's when we have a disease out there. And now one more carcinogen is there, biological carcinogen. What do you mean by that? That means in our cells, in our DNA itself, there are certain regions which does the job of something. They help in what you call all the DNA functions out there. But then if they are inactivated as of now, they are inactivated usually. If they get activated because of some mutations out there, they become cancer causing. They cause cancer, my dear children. They will make proteins that can cause cancer out there. And these genes which are there, they are known as proto-oncogenes. Okay, what do you call it? Proto-oncogenes. Oncologist is what do you call a cancer specialist, my dear children? So proto-oncogenes, or similarly cellular oncogenes, whatever you want to say. So physical carcinogen, ionizing radiations like X-rays and gamma rays. Ionizing radiation means they will make the ion being released from the atoms out there, my dear children. So they are going to cause a lot of trouble up there. So ionizing radiations like X-ray and gamma rays, and non-ionizing radiations like UV radiations can be physical carcinogens. Chemical, tobacco smoke, industrial chemicals, all of it are chemical carcinogens. Biological carcinogens, the viruses, that is also there. There are certain viruses that can cause cancer. So viruses causing cancer called oncogenic viruses, okay, they would be having oncogenes within them. They would be having certain genes within them which are transferred to us, which is why we also have cancer. Okay, so these cancer causing viruses are known as oncogenic viruses. Now several genes called cellular oncogenes, c-onc, or proto-oncogenes have been identified in the normal cells, which when activated it can cause what? Cancer. That is also there. Okay, done? Deal? So next thing, now we are entering the 5-marker session, my dear children. So we discussed enough of 3-marker questions, now the 5-marker questions. Okay, name the causative agents of the following diseases. Malaria is caused by, tell me. Ma'am, we discussed this so many times. Yes. But what should I do if questions come? So Plasmodium, Biovax, whatever you want to name, please name it. Filariasis is caused by Wuchereria, Bancrofti. And apart from Bancrofti, what do you have? Wuchereria malai. Okay. Ascariasis is caused by Ascaris. You don't need to write the whole name for Ascariasis because Ascaris lumbricoides has not been mentioned in your NCERT. If at all you want, you can write lumbricoids. Okay, amoebiasis is caused by Entamoeba histolytica. It is also known as amoebic dysentery. Histolytica. Now, pneumonia is caused by Streptococcus pneumoniae or Haemophilus influenzae. Why should I write in a bad handwriting if at all you have it over here? So, Plasmodium vivax, P. malaria, P. falciparum, Wuchereria W. bancrofti, W. malai, Ascaris lumbricoides. If you want, you can write it. Entamoeba histolytica, Streptococcus pneumoniae or Haemophilus influenzae. Then, easy now? Okay? Okay. Now, next question. What are lymphoid organs? Write the functions of the following in response to immune system in our body. This was given in the model question paper. What are lymphoid organs? Lymphoid organs are the place where the lymphocytes are being formed and they show their effect. These are what? Lymphoid organs. Now you have to mention that there are two types of lymphoid organs, the primary lymphoid organs and the secondary lymphoid organs, and then you can go ahead about talking about all of this thing. So, the primary lymphoid organs are what? Bone marrow and the thymus. Why are they known as primary? Because over here, the immature lymphocytes differentiate into what? Antigen sensitive lymphocytes. The immature lymphocytes which are being formed over here, they are going to convert into what? Antigen sensitive. Now they know their duty. They are being taught their duty. That is, you have to be sensitive to antigen. You have to kill them later on when you see it. So this is the primary lymphoid organs. Let me tell you, bone marrow is a place where all your blood cells are being formed. Your RBC, WBC, platelets, everything of it. Okay, all of the cells are being formed over here. Now in WBC, my dear children, you know there are WBCs are known as leukocyte, and in WBC there are different types, monocytes, eosinophils, this and that, and lymphocytes as well. You have B and T lymphocytes. So the B lymphocytes, they mature in the bone marrow itself. The T lymphocytes, they go to thymus to mature. And by maturing, I mean they become what? Antigen sensitive. That is what I mean by that. Now, after maturation, the lymphocytes migrate. They go to what? The secondary lymphoid organs like spleen, lymph nodes, tonsils, Peyer's patches of small intestine, and also appendix. Okay, they go to all these regions. And what do? The secondary lymphoid organs provide the sites for interaction of lymphocytes with what? The antigens. They provide the clear-cut area where the antigens and the lymphocyte come face to face and they can fight with each other, and the lymphocytes can kill these antigens. Okay, which then proliferate to become what? Effector cells. So they become effector cells. Please write this term, effector cells, antigen sensitive lymphocytes. All of them are important. Make sure you're underlining things when you're talking about them. Okay, so they become effector cells over here in the secondary lymphoid organs. Now you are been asked to explain all of them. So bone marrow, as I told you, is a primary site to produce. Where is bone marrow? Within our bones. In our bones we have yellow marrow, red marrow. In the red marrow, so bone marrow is made up of yellow marrow and red marrow. Red marrow is basically the place where your red blood cells and everything is being formed. Red blood cells, white blood cells, platelets, as I told you. So the primary site to produce hematopoietic stem cells. So there are stem cells, and stem cells has a unique property to form different type of cells out there. So hematopoietic stem cells means they are blood stem cells that can later give rise to what? Red blood cells, white blood cells, as well as platelets. And this is where also please mention that in white blood cells you have lymphocytes, in that you have B and T lymphocytes. So the B lymphocytes are going to get matured over here. Please write that. B lymphocytes are matured over here. Okay. Now thymus, an organ where T lymphocytes mature. The next type of lymphocyte, that is what? T lymphocytes. The thymus, that's why the name thymus is there. Thymus is more towards your sternum region, at the topmost, not the topmost region. Behind your sternum, you have what? The thymus being located. Okay. Sternum is a point where, this is also known as chest bone, through which all your ribs are being attached. Okay. So yes, an organ where the T lymphocytes mature. The thymus is active in childhood and early adolescence, but shrink after the puberty. After the puberty, they are going to shrink in size. Now spleen is there. So both of them were primary lymphoid organ. Now you have spleen, an organ that filters what? Okay, so I'll tell you what is the usual function of spleen. Spleen is also known as the graveyard of RBCs. Why a graveyard of RBC? Because ma'am, over here the RBC is basically die off in this particular place, okay, in the spleen. So what exactly happens over here? The worn out, after 120 days, the RBCs, they die off, okay. So the RBCs come up over here in the spleen, the worn out and the things like that, they have been caught up by all the other white blood cells and they have been broken down, my dear children. So while doing so, okay, the lymphocytes are also there. Okay, the lymphocytes are anyway in the spleen, my dear children. They will be coming in contact with certain antigens also in the spleen, and now they are going to kill the antigens as well. So an organ that filters blood, removing old or damaged red blood cells, it also helps in immune responses by storing the white blood cells and acting as a site where immune cells can detect and respond to what? Pathogens, which I call as antigens. Now there is one more thing, that is lymph nodes. So lymph nodes, I told you lymphatic vessels are there, you remember? Lymphatic vessels are the one which carries lymph fluid between the cells, the capillaries, the leak, and now you have plasma and certain white blood cells, okay. Plasma and certain white blood cells as well as certain things which is there in the plasma, they also move out over here, including the pathogens. So now they are in between the cells, in between the normal cells of our body. From there, it has been taken up through the lymphatic vessels. Now you know what? Over here, there is like a bulge sort of thingy. Again, it will go on like this. These are the places where you have so many lymphocytes already been there. Now what do? Over here in this particular lymphatic vessel comes the antigen. These antigens are going to be trapped up over here because at the lymph node, these places are known as lymph nodes. At the lymph node, what is happening? There are so many lymphocytes and they're gonna kill them. Which is why when we go to doctor, sometimes they check here. If at all there is some infection. Why? Because here also you have a lymph node. If they are swollen, that means yes, there is some problem happening in your body. Okay. So I hope that information is too much information for you. So small bean-shaped structures that filter the lymph, that is a fluid that circulates through the lymphatic vessel. They trap foreign particles such as pathogens and are key sites for immune cell activation. That means the immune activity. That's all. Easy. Don't make it complex by big English terms out there. Use simple language and score your marks. Okay? Now, 37th question. Explain the different techniques which help in cancer detection as well as diagnosis. So what are the different techniques that are useful in cancer detection as well as diagnosis? First one we all know, might have heard, biopsy and histopathological study. What do you mean by biopsy? So if there's a small, what do you call, lump out there. Okay, let me first take all of them to be here. Okay. Suppose there is a small lump out there. So what I'm going to do in biopsy, I'm going to cut a small part of it. Okay, small part of it, and then I am going to screen this. I'm going to screen what? Screen this tissue out there. This process of cutting and removing this is known as biopsy, and later checking it is known as histopathological study. This is known as histopathological study. So first technique is that. Second, we all know there is CT scan, okay, computerized tomography, my dear children. Over here, what do you use? What type of rays do you use? You use X-rays. And this is also the best way to see the internal organs out there, CT scan. But since you are using X-rays, and X-rays are ionizing radiation, is not considered to be very much safe. So what do you have as a safer option for you guys? MRI, magnetic resonance imaging, right? Magnetic resonance imaging, where basically over here you are going to use the magnetic fields and the non-ionizing radiations. This is also used to accurately detect the pathological and physiological changes in the living tissue. So if at all you want to access the living tissue out there, you can use MRI. Okay. So here magnetic fields are being used and non-ionizing radiations are being used. In computer tomography, X-rays, which is ionizing radiation, that is being used. But you also should know what is the use of it. Question comes in MCQs also. What is that thing that is used for a three-dimensional image of the internals of the object? Internals of the object, what is it? Computer tomography. Which is that something that is used for pathological and physiological changes in the living tissue. It is what? MRI scan. So these are imaging techniques. And now you have one more thing. Antibodies against the cancer specific antigens. suppose we know that in cancer, these are the antibodies whose level goes high. Okay, we know that already, we have a pre-notion of that. Now, if at all there is a suspect, there is a person who has cancer and we are yet to decide if they have cancer or not, we want to check it. So, in their blood, if the same antibodies level is high, what do we get to understand? That yes, there are chances that this person can have cancer, which is why they had the antibody specific to it. Antibodies are not released randomly out there until and unless there is some other problem out there in the body. You get it? So, antibodies against cancer specific antigens are also used for detection of certain cancer. You do not give them antigens, please remember. You're just going to take their blood and see if at all there is elevated level of some antibodies which are usually produced in cancer patients or not. That's all. Now, apart from this, techniques of molecular biology can be applied to detect genes in individuals with inherited susceptibility to certain cancers. Now, genes can also be examined, but again, that's a very long process if at all it is done. Okay, so these are certain techniques that you need to remember. Biopsy, histopathology, and in biopsy, a piece of the suspected tissue cut into thin sections and stained and examined under what microscope, that is known as histopathological studies. And then there are imaging techniques, as I told you, and then you have antibodies, and then this. Done? Cool. Next question. Some drug bottles had their names labeled. This is a model question paper question, and I love this question somehow because it's like it makes you feel like, oh my god, I'm a doctor. So some drug bottles had their names labels missing in the drugstore of a hospital. So there were some bottles, drug bottles as in medicine bottles, but the name were missing. So the staff needs to identify the drugs with their actions still written on them. So the actions are still written, but the label is missing. What as an action, as in what do they do? So analyze their actions listed below and identify the name of each drug and also the source of each one of them. So you have to tell the name and also the source also. Where do you get that particular drug from? Okay, so if at all not the source, at least the name of the drug would be fine, I think. Used by doctors as sedative and painkiller. Drug 1. Sedative and painkiller used by doctors also. It is morphine. Where do you get this from? Where do you get morphine from? Papaver somniferum. First one. Okay. Drug 2. Let me tell you, those of you who are able to identify all the name of the drugs, you have to comment down in the comment box that, man, I identify all 5 of 5. I'll be really happy to know that, and it's a really fun activity also. I don't know why, but it's fun. So second one, helps patients to cope with insomnia and depression. Insomnia and depression, remember sleeplessness, amphetamines, benzodiazepines, you can write that, barbiturates, amphetamines and benzodiazepines. These 3 things you can write. The source of it is not being mentioned in your NCERT, so you don't need to write it. Now drug 3, increases the blood pressure and heart rate of consumer. Have any idea? Ma'am, the one that helps in the cardiovascular system. No, not that. Blood pressure, whenever the term blood pressure comes in, just remember this thing, smoking. In smoke, I told you nicotine is there in the tobacco, and nicotine is something that increases the blood pressure. Always remember. So it's nicotine. So increases blood pressure and heart rate of the consumer, it's nicotine. Act as depressant and slows down body function. I told you there's only one, heroin. Okay, it is what? Heroin. That is going to act like a depressant. Now, affects the cardiovascular system of the body. It can be charas, it can be ganja, it can be hashish, it can be marijuana, anything that comes under cannabinoids, and you get it from cannabis sativa. So for those of them whose name is given in your NCERT as in the source plant, you can put that. Otherwise it's okay. Okay. So morphine, barbiturates, amphetamines and benzodiazepines, nicotine, heroin and cannabinoids. Okay, and morphine you get from papaver somniferum, nicotine that's not given, tobacco plant basically. Okay, so over here heroin, again papaver somniferum, and over here cannabinoids, cannabis sativa. Fine. Now I think this is the last question for the day, which is, give the schematic representation of replication of a retrovirus. So where do you study this? You study this in HIV virus. How does that particular virus replicate in our body? What exactly happens over here? So if at all you have to give the schematic representation, how do you make it, my dear children? So over here, let me take you to right over here you have the retrovirus. So retrovirus has a viral RNA code. That means it has RNA, does not have DNA, it has RNA. Let me tell you, it has 2 strands of RNA, one strand, another strand. So ma'am, it is double stranded? No, they are single stranded. Both of them are individual, but they have 2 strands. Remember that. 2 single stranded RNA is there. Now it is covered by a protein covering. You call this as what? Capsid. What do you call it? Capsid. Okay, then that's there. So protein covering is there, and the viral material, the genome is there. Now virus infects the normal cell. First question, which is the cell that it initially comes and affects always? It is also known as the HIV factory. I am not talking about T helper cells. T helper cells are something that it is going to gradually affect and going to destroy them totally. That is there. The most affected one is T helper cell. But before that, they come and replicate, they increase the number in one type of cell which is known as macrophages. Okay, please remember. It is the HIV factory, it is known as macrophages. So what exactly happens is that these viruses, they come over here, they have certain proteins on the top of their membrane, my dear children. Slowly these proteins interact with the proteins on the cell surface as well, and they get inside. You just have to understand that they are going to transfer their genome, the RNA, into our cell. Okay, inside our cell we have their RNA. Now, using an enzyme which is known as reverse transcriptase. What is transcription? We studied it. All of you have studied molecular basis of inheritance with me, knows what you mean by transcription. DNA to RNA is known as transcription. Now if you want to reverse it, what will it be? From RNA you form the DNA. Same thing is happening over here. But the help of reverse transcriptase, the RNA which is there, it will help in the formation of what? DNA, and that too a double stranded DNA out there slowly. Okay. Now this double stranded DNA which is there, which is being formed, so it has all the information of what? Of the viral genome. As in what proteins does this particular thing need to be formed, and what are the other things, and everything, whatever is a major problematic things, all the information is stored up in this particular DNA. Now what is the purpose of converting it into DNA? So that it can get inside our cells nucleus and then get incorporated into our genome, because we have a DNA genome. So the viral genome, my dear children, with the help of another enzyme which is known as integrase, okay, with the help of another enzyme that is known as integrase, gets integrated into our genome. Fine. Now they are inside our genome. Now what happens, my dear children? RNA is being formed. We all know here, after the DNA is being formed, transcription happen, RNA is being formed, and this RNA is going to give the information of what proteins have to be made, right? So whatever proteins have to be made is made. So protein covering is there, RNA genome we already have got from the DNA. Okay, so everything has been done. Now you have so many viruses out there, they just have to move out. That's all. So this is how you give the schematic representation. Okay, done. It's easy. So there are 3 enzymes actually: reverse transcriptase R, okay, integrase I, and there is P also, protease. Okay, not protease, it was protease or permease. Just a second, okay. Yeah, it was protease itself, as I initially told you. So they are there for cutting the the long chain of proteins being made into smaller fragments, so that it can be used up for making a whole viral thing out there. Okay, so these are the 3 enzymes, RIP, rest in peace. So this is the thing, you can remember like that. So these are the enzymes, and this is how you have to put it down. So whenever you write it, make sure you're writing 1 point: virus infects the normal cell, and then viral RNA is introduced, then viral DNA is produced by reverse transcriptase, then viral DNA incorporates into the host genome, right? Point by point, point 2, point 3. Here point is not mentioned, but I'm telling you. New viral RNA is produced by the infected cell, and the new viruses are produced, and then, my dear children, they move out. New viruses can infect what? Other cells. Now these other cells are what? The T helper cells out there. So this is something that you need to remember. And with that, what we have covered all the very important questions of this particular chapter, that is human health and diseases. And with this, I can guarantee you are getting whole 11 on 11 marks of this particular chapter. Okay, if you think that this video was useful for you, you have to let me know that. If you have any doubts still persisting in your head, put it down in the comment section. Listen, I am not here to judge you. I will never judge you. So if at all, whatever silly doubts you are having, you can put it down. I will tell you the answer for it, and you do not have to be worried about anyone else who is seeing the comments. No one knows you. Okay, so please put it down. I'll help you. And my dear children, as I said, if at all you think that these videos that we are putting are useful, what you have to do, share it with the friends who are in need. Okay, so that's all. Wishing you all the best. Lots of love from Jasmine ma'am. Bye-bye. Take care.